Hello, and welcome to part two of our lecture series on the integumentary system around the skin. And in part two, we're going to continue on our overview of the epidermis, the outermost layer associated with the skin. Now, in the previous lecture, in part one, we looked at the formation of, in essence, the maxillary keratinized dead cells, the cells along the outer surface of the skin within the stratum corneum. And so again, keep in mind that what we've got is cell remnants that have a lot of keratin within them. They're maxillary keratinized, but it's essentially cytoskeletal elements that are going to be cross-linked. They're going to give a lot of strength and integrity to the outer surface of the skin, so that when you brush up against something, you're not leaving body parts behind, because these cells, even though we may slough off the outermost layer, there's still going to be cells underneath, and it's got a lot of resistance to friction and a lot of resistance to abrasion. One of the other properties associated with the epidermis is going to be the fact that we've established a waterproof boundary within the skin. So again, keep in mind that the surface cells, the cells in the stratum corneum, are exposed to a very dry, arid external world. And essentially the cells out there are dead. But inside of, say, the levels of about the stratum lucidum, we're getting cells that have nuclei, cells that are going through biological processes. They're alive, they're doing things, they're producing the keratin, in essence, the cytoskeleton. If we go deeper, we've got cells with DNA that are continually dividing. And we've got the entire rest of the body. And so we've got this aqueous living tissue and living cells that has to be protected. And so what we're going to look at next is how the epidermis of the skin establishes that waterproof boundary. And what we're going to see is that these keratinocytes, the same cells that are becoming maxillary keratinized along the way, they're also going to be producing what are called membrane coating granules or lamellar granules. And so basically what these are membrane-bound structures. And so they're going to be produced, they're going to have a membrane limiting around them. So we got a distinct portion of membrane, and they're going to be packed with a lot of lipids and a lot of glycolipids. And so we're going to have a membrane-bound organelle within the cytoplasm. We have lots of lipids in it, packed within it. And as these cells move up within the epidermis, as basically it's forming within the stratum spinosum, going up through the stratum granulosum, and as we reach those upper levels of the stratum granulosum, these membrane-bound granules, these membrane-coating granules or lamellar granules, are going to fuse with the plasma membrane and essentially through the process of exocytosis dump out their lipids. And so if we think about the properties of lipids, lipids are hydrophobic. Lipids, like the lipids within the plasma membrane, are going to be resistant to allowing water to flow through them. So by dumping out a lot of lipid into the space between these cells, between the keratinocytes, at the upper reaches of the stratum granulosum, we're essentially going to establish a lipid-rich, or essentially fatty layer within the skin, which is going to block the movement of water. Water isn't going to move through this fatty hydrophobic region and run the risk of dehydration. So in essence, what we've got then is this ability to produce these membrane-coating granules, pack them with lipids, dump it out along the surface of the cells, going out probably about the level of the stratum lucidum, but at least a little bit towards the last portions, the highest portions within the stratum granulosum. And so what we're going to have is lots and lots of lipid packed together between the remnants of these cells, because as we're going through there, the cells are going to be surrounded by the lipids, and so they're going to start to essentially be isolated from the rest of the cells. And as they push higher into the epidermis, so they get closer to the surface of the skin, they're going to get through this waterproof boundary, and they're essentially going to dry out, and they're going to, in essence, go through a programmed cell death. And so we're going to have this barrier there. It's going to be important for blocking water. The other thing is that it's going to be very good for blocking the entry of foreign substances. So materials that are on the surface of the skin aren't, under normal circumstances, going to be able to diffuse from the surface of the skin into the body, because they've got to pass through this lipid barrier. There's still some things that do that, but in general, bacteria and other harmful agents are going to have a hard time penetrating through this lipid-rich layer that's going to be present within the skin. Okay, and that's all I'm going to say about the keratinocytes right now. Those are the majority of the cells. They're the cells that produce this maximally keratinized stratified squamous epithelium. The next cells we're going to look at are going to be the melanocytes. Now, the melanocytes are going to be relatively common cells, but much fewer than we're going to see the keratinocytes. The melanocytes are going to be sitting in among the keratinocytes within the stratum basale. And essentially you can identify them because they're going to have a relatively pale cytoplasm. And so you can take a look at it, the image on the right-hand side of the slide. You can see the nucleus, a little bit euchromatic nucleus, and then a pale staining cytoplasm around it. That's an example in that central location sitting there on the stratum basale of a melanocyte. And now what's going to happen is the melanocytes are basically going to produce melanin. They're going to produce a pigment, which is going to be a protective mechanism. They're going to block ultraviolet radiation from penetrating the skin and potentially damaging the DNA of the cells below it. So what's going to happen is the melanocytes are going to produce the melanin. They're actually going to pack it within melanosomes, and these melanosomes are going to be transferred to the keratinocytes. And so in kind of a weird juxtaposition, the melanocytes, the cells that produce the pigment, are going to be pale, and they don't accumulate the melanosomes. They don't accumulate the melanin, they don't accumulate color, because they're essentially taking those properties, taking the melanin, and inserting it into the neighboring keratinocytes. Okay, so if we take a look at this, the melanosomes are going to be another example of a membrane-bound structure. And so essentially we're going to be in the production of the melanin. We're going to be going through a chemical process. We're going to convert tyrosine to dopaquinone into melanin. And so we're going to see enzymes such as tyrosinase, which is going to be important for converting tyrosine to dopa. And basically, this is going to be occurring as we're producing the melanin. And so, in essence, what we're doing is we're going to be synthesizing it. We're going to be packing it within the Golgi, putting the mature form within these melanosomes, within these membrane-bound structures, and then ultimately taking these membrane-bound melanosomes and getting them into the keratinocytes, where they can be accumulated above the nucleus of the keratinocytes and protect those cells from the effects of ultraviolet radiation. Certain forms of albinism, essentially individuals that don't have a color associated with their skin, is associated with the absence of the tyrosinase activity, the absence of the enzyme involved in the production of melanin. Okay, so again, what we're looking at is the idea that this melanocyte is going to be sitting within the stratum basale. It's going to be producing melanosomes, so producing melanin packed within the Golgi, packed within a membrane-bound structure within the melanosomes. And then the melanocytes, these cells, are going to have cytoplasmic extensions. They're going to reach out and through in a cytokine mode of secretion, so essentially do cell-to-cell transfer. And so they essentially pinch off a little bit of their cell membrane, a little bit of cytoplasm within it, but lots of melanosomes. And those melanosomes are essentially then either injected directly into the keratinocytes or they're phagocytized by the keratinocytes. The different books say different things. But the important thing is that they're essentially transferred from the melanocyte, the cell producing the melanin, into the keratinocytes that are going to take those melanosomes, these membrane-bound granules with the melanin above it, essentially move this above the nucleus, and it's going to have the effect of preventing ultraviolet radiation from penetrating the deeper regions of the skin and potentially damaging the DNA. It's often also been learned relatively recently that the melanin may have some antioxidant effects as well. And so it may be able to go through and essentially quench some of the free radicals that may be formed in response to ultraviolet radiation. If we take a look at an individual skin color, what we can see is that there are a number of factors that are going to be contributing to it. Things like the melanin that we're talking about right now are going to be a dark brown color, but there are things like bilirubin, which is a byproduct of degradation of hemoglobin in the liver, which contributes to kind of a yellowish brown appearance. Hemosiderin is kind of a golden brown pigment, again related to the breakdown of hemoglobin. Things within foods, carotenes, the diet, in say rich orange vegetables, looking at like squashes or carrots, things like that. Oxyhemoglobin may contribute to kind of a reddish hue to the skin, again because of the presence of blood vessels relatively close to the surface of the skin. If we take a look at the differences in skin color kind of at one level, we can take a look at melanin production and melanin breakdown. And basically what we see is that in Caucasoid skin, we can have a number of melanosomes that are taken up by the keratinocytes that are within these cells, but they're basically going to be broken down relatively rapidly, again, through that process of intracellular digestion, where the melanosomes are essentially going to be fusing with lysosomes and breaking them down. So we see the the melanosomes and the melanin are going to be towards the lower layer levels of the epidermis and broken down relatively rapidly. In contrast, in negroid skin, we can see this situation where we're producing generally one melanosome per vacuole, per membrane-bound structure. It's going to get into the cells, and there's a lot less lysosomal activity. And so because of that, these melanosomes and the melanin essentially stay around longer within these cells, and it is present within these cells at a much higher level within the epidermis. We take a look at the last two types of cells. Again, the Langerhans cells and the Merkel cells are relatively rare cells within the epidermis, but we're going to talk about them. The Langerhans cells were identified about 150 years ago as a stellate, kind of star-shaped cell, pale staining within the stratum spinosum, and in essence, you can't identify them reliably with standard light microscopy. But they're an example of an antigen-presenting cell. So they're going to extend throughout the epidermis. They're going to be picking up particles of things as they're kind of reaching through it. And they're going to transmit that information, transmit these shapes, in essence, the potential antigen, to other cells. And in doing that, essentially, hopefully jumpstart an immune response if there are foreign materials or disease-causing materials that are passing through the skin. The Langerhans cells are going to be identified by kind of ping-pong-shaped membrane-bound cytoplasmic granules called Burbitt granules within their cytoplasm. But again, they can only be reliably identified using transmission electron microscopy. The last type of cell within the epidermis are going to be the Merkel cells. And again, these are going to be very, very rare cells found essentially within the stratum oculi, so sitting along the basement membrane, and they're going to be found in areas of very high sensory perception, so like the fingertips. Basically, these cells are thought to be touch receptors because they have a nerve terminal that extends on them. And so these Merkel cells are in contact with sensory innervation to the skin, and because of that, they're thought to have some type of sensory function. And this finishes up our look at the epidermis. Essentially finishes up part 2 of this lecture series. In part 3 of this lecture series, we're going to take a look at the dermis, or the connective tissue components that are sitting underneath the basement membrane that our epidermis is sitting upon. As always, if you have any questions, feel free to email me at hoffmanj@arcadia.edu.